Hi everyone, this is our experiment 1. I'm going to help you with your pre-lab assignment. Let's see what we are supposed to do for our experiment 1. We're going to find out rates of a chemical reaction. We're going to find out the rate constant and the reaction order with respect to individual reactants. I believe by this time you may have studied rates of reactions in your lecture. But in this experiment, we are going to put it into practice, what you have learned in your lecture. Okay, so the particular reaction that we are going to study is this one that you see as reaction 1, where we're going to be using iodide in terms of potassium iodide, potassium bromide in the presence of hydrochloric acid, that's the your HCl, H+, giving you this iodine I2 gas. I2 it's going to be dissolved in water, so it's going to be aqueous, plus potassium bromide and water. So in this particular reaction, we can study the rates with respect to individual reactants. So our reactants are going to be iodide, bromide and H+. So you can see in equation 2, we can write the rate. You know K is rate constant, and then the concentration of I minus, that could be the order M. Okay, we can consider that as order M. With respect to bromate, order could be M, N, it's going to be N, and then with respect to H plus, hydrochloric acid, that can be order P. So this M, N and P, they are the orders of reactions. That is going to be different than the reaction coefficients. Now you see in equation 1, there are 6 I minus, 1 bromate and 6 H plus. So those are your molar ratios. They are not the same as M and P orders. So the order will tell us by changing the concentration of the reactant how the rate is going to change. Is it going to be the 0th order? 0th order means no matter how much you increase the reactant concentration, order doesn't change. But if it will be first order with regard to a one particular reactant, if the rate doubles, and it's going to be second order if it is going to be quadruple the rate of the reaction. So M, N and P, they are going to be reaction orders. So in this particular reaction, we are going to find out what is the rate constant as well as what are the orders M, N and P. Okay. Now at the same time, we are going to make another reaction take place as our main reaction is going on. So the other reaction is we are adding some sodium thiosulfate, S2O32 minus, that's the anion, so sodium thiosulfate in the presence of the iodine that generate is going to form a complex. So iodine is going to react with sodium thiosulfate. So for each reaction mixture, we're going to be adding a constant amount of sodium thiocyanate. So thiosulfate. By adding that, we introduce a clock to the reaction. So once all the thiosulfate ions are reacted with the iodide generated from the reaction, we have another indicator in the mixture, that is a starch indicator. So the iodine that is going to produce going to react immediately with the starch and it gives us a blue color. So what we do is, as we mix these three reagents, your iodide, bromate and hydrochloric acid, we start a clock. And then we're going to monitor the reaction. You start your stopwatch and then we're going to see when you see the blue color, and then you write down the number of seconds that it takes for the reaction mixture to turn blue. Okay, so likewise we're going to be conducting 4 or 5 experiments using different amounts of the reagents. Okay, so that's what we are going to do. Excuse me. Now the experimental procedure you can find here. So this is the table how we are going to mix the reactions. So you can see there are 5 reaction mixtures that we're going to be doing, 4 or 5. Okay, sometimes we do 4, sometimes we do 5. Let's see how we are going to mix these reagents. So you basically going to take 2 flasks. Okay, so you will see in your virtual lab you will have two reaction flasks. Reaction flask 1 and reaction flask 2. So for the first flask, you're going to be adding potassium iodide, your sodium thiosulfate and water. And for the second reaction flask, you are adding potassium bromide and hydrochloric acid. Okay, so what you're going to do for the reaction mixture 1, you are adding 10 milliliters Ki, 10 milliliters sodium thiosulfate, 10 milliliters water. And for the second flask, 10 milliliter potassium bromide, 10 milliliters hydrochloric acid. So you can see for all the reaction mixtures, we're going to be keeping sodium thiosulfate a constant amount, 10 milliliters. Now what's going to be the total volume of the reaction mixture once you mix these two reagents together? 10 plus 10 plus 10, 10. So 10, 20, 30, 40, 50. So reaction mixture, the total volume of the mixture is going to be 50 once you mix the two flasks together. Okay, so the first flask goes the first three reagents, second flask goes the second two reagents, and also for the second flask you are adding starch. That's how we're going to perform the experiment. Okay, and for the second reaction mixture, you are adding 20 Ki, 10 thiosulfate, no water, 10 potassium bromide and 10 hydrochloric acid. So what is the total volume going to be? 20 plus 10 plus 10 plus 10, 50. So if you look at all 5 reaction mixtures, the total volume is going to be 50. Okay, we need to find out what are the concentrations of each reagent of our reactants in this 50 milliliter sample. Once you mix it together, it becomes 50, right? So when you put flask 1 content and flask 2 contents together, your reagents that you are taking, even though you are initially potassium iodide 0.01, but now the concentration is going to change when it is in the 50 milliliter mixture. So we need to figure that out. Okay, we can do that using the dilution equation. It also explains how we are going to mix. So it's just like what we see in the table, and then you will have the experiment data part. Okay, data and the calculations. So we can see how the data reaction, the main reaction is given here, and then you have to complete this table, okay, in your data sheet. For reaction mixture 1, time for the color to change when you mix the two together, how long it take for your blue color appear, right? It could be 75 seconds, 40 seconds and so on. You're going to write it down. Okay, let's say for the first one, if it takes 40 seconds, you're going to write it down. And then second one, 75. And then the relative rate of the reaction. So the rate is going to be inversely proportional to the time. So once you get that 40 seconds, what you're going to do here is 1000. You use 1000 over 40. And then you write it down, the number right there. 1000 over the time, this time. So likewise, you will get the data from your virtual lab. You're going to do 1000 over 40 and you write down that numbers over here. Also, you need to find out what are the reactant concentrations in your 50 milliliter sample. To do that, you can use this dilution equation. We know that M1V1 equals M2V2. So the initial concentration of iodine stock solution is 0.01. Okay, it's given to us in our table, 0.01. And then how much are we taking from our stock solution? For the first reaction mixture, you take 10 milliliters. And then the total volume, once you mix the flask 1 and the flask 2 contents, the total volume is going to be 50 milliliters. Okay, let me show you this, where this 0.01 come from for potassium iodide. It's right here. You see Ki 0.01 molar. That's a stock solution. And you take 10 milliliters from that. And the total volume is 50. And now we want to find out, when everything is in that 50 milliliter sample, what is the concentration of Ki going to be? You calculate that using this dilution equation. So M1 0.01, V1 10, V2 50. So then M2 we can find out 0.01 times 10 over 50, 0.002, and that is the number that you're gonna write down over here. So the first one is done for you. Similarly, you have to calculate what is the concentration of bromide in the 50 milliliter sample. So you refer to the table, your table potassium bromate. You go over here, you find out now the potassium bromate over here 0.04, and for the first reaction mixture you take 10. So M1 going to be 0.04, V1 going to be 10, and then the V2 going to be 50, and then you can find out the M2, what is the concentration of potassium bromate. Similarly, you need to find out the concentration of hydrochloric acid in your 50 milliliter sample. That's for the reaction mixture 1. And then for the second reaction mixture also, depending on the volume you take, you have to calculate. So likewise, you can go ahead and complete this reaction table. You need this for your post-lab calculations. And for your pre-lab assignment also, you're going to be using similar calculations. Okay. So once you get these data, then we can process them to find out the orders of the reaction. Okay. Now with that in mind, let's go ahead and see how to calculate the questions in the pre-lab assignment. Okay. So the question number 1, in no more than 2 sentences, describe the objective of this experiment. Well, you can do that. It's a written answer choice. So you can write in your own word what is the purpose of this reaction objective. Question number 2, if you mix the reagents for each reaction, what is the total volume of the solution in each of your reactions? So what is the total volume? Remember, we talk about that. That's going to be, what is it? When you add the contents in the 2 flasks, it's going to be 50 milliliters, correct? Type your answer, express in units of milliliters, 50 milliliters. And then question number 3. Given your answer to question 2, what is the molarity? What is the molarity of iodide in reaction mixture 1? Okay, so you will be using 0.01 molar Ki. So you can find out what is the concentration of iodide. So we can use M1V1 equals M2V2. So the concentration of potassium iodide 0.010, and then the volume from the table that I have showed you for the reaction mixture 1, you will be using 10 milliliters. And then the M2 you don't know, and then the total volume is going to be 50 milliliters. So when you do that calculation, you can find out the M2. Okay, do the proper calculation and check to see if you are getting 0.002 molar solution. Okay, express your answer to 2 significant figures. So then it should be 0.0020 molar. Excuse me. Okay, and then if you are going to express in exponential notation, it should be 1, 2, 3, 2.0 e negative 3, like that. Okay, and question number 4.